To NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology master teacher. I hope I'm audible, visible, and the presentation is also visible. Please let me know in the chats if we are audible and visible, and are we live yet? I think yes, we are live, right? Just checking the audio as well, students. Let me know in the chats if we are audible and visible, and are we live yet? Okay. Perfect. So today we are going to do neural control and coordination. As all of you know that we have been doing human physiology, and we have finished breathing, we have finished excretion, we have finished locomotion, and locomotion also once so sorry locomotion also one small portion is left, and that I'll be finishing soon. Okay. So today we are going to do chapter 21, that is neural control and coordination. As you know, the functions of organ and organ system in our body must be coordinated to maintain homeostasis. What is homeostasis? That means the balance in the body, okay? All the functions in the body and everything that happens in the body goes through a balancing. Okay, so if we have to balance the body temperature, if we have to make sure that everything is coordinated, we need some system, right, who is going to do all the coordination. So that is going to be your neural control and coordination. Now students, since we have to study chemical coordination, we all know that we have endocrine gland also, right? We have endocrine gland also, and this endocrine gland also is going to do something called as coordination. Why is it going to do something called as coordination? Because they are releasing hormones, and those hormones have some functions, right? These hormones reach the target organs, and they are going to do one set of functions. So same thing is going to happen here, but here there could be a difference when we talk about neural control and coordination and endocrine gland, correct? Because endocrine gland releases their hormones, the hormones goes to the target organs, and the target organs are going to perform a function. Let it be growth hormone, let it be anything. Same way, now your neural system also, what is going to happen? Now let's imagine this cup that I'm having is a hot object. Okay, inside this is a hot object. The moment I'm going to touch this object and I realize that it is hot, the signal will already be sent to our central nervous system or to our brain, telling that Gopika touched a hot object, correct? So immediately my hand is going to retrieve from that object. So what does it mean? It means that my neural systems are working faster. And see, it is going to work for a short duration, because that is the only signal that, okay, Gopika has touched a hot object, please retrieve her hand. This is the signal. So it is not going to be longer as much as in endocrine, but in endocrine it's going to be longer because the hormone secretion is going to happen, the target organs are going to work according to it. Okay, chalo. Coordination is a process through which two or more organs interact and complement the functions of one another. Okay, so coordination means what, students? Now imagine I am running a show. Okay, I am running a fashion show, and in that fashion show I will make sure that all my models are ready. Yes or no? So that will be the first step. All the models are ready. Then I will make sure that all the dresses are ready, hairdresser, whatsoever. So many people are behind it. All of them are ready. Why? Because everything has to get coordinated. Everyone should interact with each other, and one should complement the function of another. That means if, let's say, a makeup artist is there, and let's say I hired that makeup artist only for 2 models, and she's like, no, I will not do for the third one, or I will not help, or if I see some error I'm not going to correct. That means that the show will not run properly. Same thing happens in our body. If something has to run properly in our body, that means everything should go through a coordination. Okay, so coordination is the process through which 2 or more organs interact and complement the functions of one another. For example, when we do physical exercise, the energy demand is increased for maintaining an increased muscular activity. The supply of oxygen is also increased. Yes or no, students? When you're doing exercise, let's say when you're running a marathon, or when you're running on a treadmill, you actually lack oxygen that time, so you will want more oxygen. So all the body muscles, let's say breathing and extend, that means your locomotion and movement, breathing and extend, all these people are going to work in coordination. Yes or no? So here see, the increased supply of oxygen necessity and increase in the rate of respiration, heartbeat, and increased blood flow via blood vessels. So that means basically all the chapters of your human physiology is going to work here, right? Breathing is going to happen, that means we need more intake of oxygen, that is going to happen. At the same time, lot of blood flow we will need so that more oxygen can be transported. Body body fluids and circulation is happening. Your muscles are going to get the signal, so they will also start contracting and relaxing faster. At the same time, students, here you can see that the heartbeat is going to be increasing. So all this, so during a physical exercise, you can imagine that what and all is happening in your body. Okay, what and all is happening in your body, and each of it has its own job, telling that okay, I will be doing this, I'll be doing this. Okay, now when physical exercise is stopped, the activities of nerves, lungs, heart and kidney gradually return to their normal conditions. Okay, thus the function of muscles, lungs, heart, blood, kidney and other organs are coordinated while performing physical exercise. In our body, the neural system and the endocrine system jointly coordinates and integrate all the activities of the organ so that they function in a synchronized fashion. I hope this is very clear. Hi, sweetie Chauhan. Isn't this clear? That whenever, let's say, anything to do with your systems, everyone works in coordination. They're not like, oh, I will not work for you, you don't work for me, that day you didn't talk to me, so I will not work for you. That kind of complex ego they do not have. Okay, so the neural system provides an organized network of point to point connection for quick coordination. Okay, now students, all of you know, if a neural system is there, let's say this is the axon, this is the ending. Okay, this is the terminal ending. Okay, this is the, it might look like a root, but understand that I'm using the pen, I cannot draw that well. So this is the axon ending, and then there will be a connection. So let's say here there is a neuron and there is a dendrite connection. Okay, this is a dendrite, so it's always head to tail connection. So you can imagine that this is a point to point connection, right? Point to point connection. Clear or not? Okay, what I'm writing is visible, right? Let me just check. It's visible. Okay, fine. Now the endocrine system provides the chemical integration through hormones. In this chapter, you will learn about the neural system of human, mechanism of neural coordination like transmission of nerve impulse, impulse conduction across a synapse, like a physiology of reflex action. Now, here. Neural system. Okay, what is neural system? The neural system of all animals is composed of highly specialized cells called neurons. All of you know neurons. Okay, neurons are the structural and functional unit of your nervous system. Okay, like how we studied about nephrons, how we studied about Malpighian tubules, right? All this when we have studied about neurons, nephrons, cell, all this we tell they are the structural and functional unit. Why do we say that? It's because students, they are going to constitute our entire system. Okay, they are the ones who are going to constitute the entire system. Clear or not? Yes? Okay. These neurons, which can detect, receive and transmit different kinds of stimuli. The neural coordination is very simple in lower vertebrates. For example, in Hydra, it is composed of a network of neurons. The neural system is better organized in insects where a brain is present along with a number of ganglion. Now, when I taught you cockroach, I have spoken about supraesophageal ganglion, right? Supraesophageal ganglion, subesophageal ganglion. What are they? They are just a group of neurons, okay? They're just a group of cell bodies who are going to do some function, telling that, hey, you know what, I'm going to take up the job of the brain and I'm going to do this function. That kind of function is going to happen. So number of ganglia and neural tissues. The vertebrates have a more developed neural system. Now next topic. So neuron is clear, no? It's a structural and functional unit, and obviously we have to draw the neuron, but I don't think in this pen it will be possible. But let's say students, a neuron, okay, neuron is there. It actually has two major portions. Okay, one is going to be a long process, and one is going to be short branched. Now look here, if you have to I think I'll just write these two together. See, okay, so students, here I will do two categories. Oh, this is too thin, no? I'll do two categories. One is going to be long process. Okay, long process, visible, right? And this is going to be short branched. Here I'm going to write soma, cyton. Okay, soma, cyton, cell body, many words are there. Okay, perikaryon, all these words are going to indicate the cell body. Now long process is going to be something that is long, unbranched, that is going to be none other than your axon. Okay, short branched is going to be your dendrite. Short branch is going to be your dendrite. So this is how your neurons are going to be categorized. Okay, so if we draw a normal neuron, let's say a Kuti neuron, we'll try to draw here as much as possible, like this. Okay, like this. This is your dendrites. So actually this is very bad. And there's no cell body only for me to show. So just give me just 1 minute. I'm going to draw it again. Okay, you draw like this. Okay, again you take like this, you take like this, you take like this, and this way. Okay, and then you continue like this. Perfect. Okay, this is better. This is the axon. Now this is going to be the nucleus, and this is going to be the dendrite. Okay, this is going to be the dendrite. Same thing here, dendrite. Same thing here, dendrite. Okay, so now this is a cell body. Now here on the cell body, students, the axon can be myelinated or non-myelinated. Okay, axon can be myelinated or non-myelinated. So here you can see. Now let's go further and see what I wrote here. Neuron are of two types, two portions are there. Okay, two parts are there. This is going to be the cell body. Okay, this is going to be the cell body with the cytoplasm and nucleus. And when you come down, there is a long process which is unbranched, that is going to be your axon. So this is the axon which is extending from the cell body, and it is going to have the axon terminals and other things. Okay, axon terminals and other things. Then students, short branch structure, this is going to be called your dendrites. Okay, this will be going to be called your dendrites. So these are the two portions of your neuron. I hope it is clear. Yes, clear to all of you? Yes. Okay, great. Now the human neural system. Okay, we are going to talk about the human neural system. Like we have learned this in our EVS and everything. It is very simple. Okay, see, let's go further. See, the human neural system is divided into two parts. What are the two parts? Central nervous system, which is your brain and the spinal cord, CNS, and then we have peripheral nervous system, which is your PNS. Now the CNS includes the brain and the spinal cord, and it is the site of information processing and control. The PNS comprises of all the nerves of the body associated with the CNS. Now students, so what is CNS? CNS is the central nervous system. That means your brain and spinal cord is going to fall under your central nervous system. Yes or no? Okay, it is going to fall under your central nervous system. Yes. Okay. Now the CNS includes the brain, and what is going to happen in the CNS? It's a site of information, right? It's a site of information, it's a site of processing, it's a site of control. All the information that how I am going to react now, what I'm going to do further, everything depends on the central nervous system. So brain and the spinal cord. Now you have peripheral nervous system. The PNS are two types, that is your afferent fibers and efferent fibers. Now students, before we go to afferent fibers and efferent fibers, now imagine your, let's say, Sweetie Chauhan is traveling to Rajasthan. Okay, this is Rajasthan. If he or she is going to Rajasthan, they will take a transport, right? They will take one transport. Could be they would have taken a train. Okay, they would have taken a train, or they would have taken a car, right? They would have taken a car, or they would have taken a flight. Okay, they would have taken a flight. Now, in any means, they can reach Rajasthan. Now, let's say same way, if the nerves have to reach the brain and the spinal cord. there are a set of nerves that is going to run to the brain and the spinal cord. Now that nerves are called as your spinal nerves and cranial nerves. Understood? These nerves will fall under your PNS. Okay, so the nerves that are going to fall are going to be spinal nerves, and your cranial nerves. Got it? The one that is going to run to the spinal cord, the one that is running to the brain. Now, afferent and efferent fibers, let's see what are they. The afferent nerve fibers transmit impulses from the tissues, organs to the CNS. Okay, afferent means what? The one that brings. Okay, so here the afferent nerve fibers transmits impulse from the tissues, organs to the CNS. That means the person who is going to bring the signal to the CNS is going to be a afferent one. Okay, even when you read it, if you find like very confusing, ma'am, transmit, impulse, what is that? Do not get confused. Understand, the one who is going to bring all the signals from the tissues and organs, okay, to the CNS is going to be afferent. The one who brings. Afferent means the one who is always entering. Okay, efferent means the one who is going back. So efferent fibers transmits regulatory impulse. So understood, one is going to bring information to the CNS. Let's say this is the CNS. Okay, this is your tissues. Okay, tissues and organs. The one that is going to bring the information to the CNS, okay, is called as efferent. And the one who is taking the information from the CNS to the tissues, telling that what you have to do, is going to be efferent. Done all of you? Yes or no? Is it clear? Easy peasy. Okay, so efferent fibers transmits regulatory impulses from the CNS to the concerned peripheral tissues and organs. So always remember, if means the one who is leaving, af means the one who is coming in. Okay, yes. Clear? Okay. Chalo. The PNS is divided into two divisions. Okay, PNS is further divided into somatic neural system and autonomic neural system. The somatic neural system relays impulse from the CNS to the skeletal muscles, while autonomic nervous system impulses from CNS to the involuntary organs and the smooth muscles of the body. The autonomic neural system is further divided. So students, it's very simple. Autonomic neural system is going to be the one who is going to send the signal. Just give me 1 minute, okay? Yes. Just 1 minute. Okay. One second, students. Just fixing that. I hope it's visible now. Yes, perfect. Okay, visible now. Okay, I'll write the informations here so that both the slides will be able to access. Okay, chalo. So I have to tell you a few things, right? First of all, okay, the pen is it's difficult to write in the pen. That's why I'm not able to write a lot. So students, here neural, okay, or your neural system or your nervous system, let's say neural system or your nervous system if you have to talk. Okay, these systems are going to be divided into two. We already learned that CNS and PNS, right? We already learned that it is going to be divided into two, CNS and PNS. Okay, now let's just write that CNS and PNS. Now CNS is further divided into what? Brain and your spinal cord. Brain and spinal cord. Now PNS, students, is further divided actually into your cranial nerves and your spinal nerves, but here they have given afferent fibers. Okay, afferent. Okay, you cannot see if I write it to the corner. No, 1 second. I'll write it in the center for all of you. Neural system or nervous system, anything you can write. Okay, two categories is divided into CNS and PNS. That is central nervous system. Okay, central nervous system and peripheral nervous system. Done. Okay, now central nervous system is further divided into two. What is it? Brain, spinal cord. Okay, brain and spinal cord. Correct? Yeah. Now this peripheral nervous system is further divided into two categories. One is called as the afferent fibers. Okay, and one is called as the efferent fibers. One is called afferent, one is called efferent. Afferent means the person. Okay, let's just draw the diagram which we drew there only. Okay, let's say this is the tissues. Okay, this is the tissues. Tissue. Okay, tissue. Now this is a CNS. I told you these are the nerves that is going. Like I told you, if Sweetie want to travel to Rajasthan, then Sweetie can take train, bus or car or flight. So that transport, right, that transport to the brain, who is going to do? The nerves are going to do, right? That is going to fall under your PNS. So students, the signal from the tissues to the CNS is going to be taken by none other than your afferent. Okay, afferent arterioles. Okay, and from the CNS, whatever signal the CNS is going to give response, that is going to be taken by your efferent. Understood? Efferent. So this is how it works. Afferent is going to carry the signals from the tissues to the CNS, and efferent is going to give the regulatory response from the CNS to the tissues. Okay, now they are telling, okay, after this they have told that again this is further divided into two categories, that is called as the somatic nervous system and autonomic nervous system. Okay, right here you can see somatic neural system and autonomic neural system. Correct? Perfect. Okay, so autonomic nervous system and neural nervous system. Somatic neural system is going to send the impulse, okay, relay impulses from the CNS to the skeletal system. That means to the voluntary muscles. Okay, to the voluntary muscles. So here somatic nervous system is going to send the signal, okay, or we can tell the impulse relay from CNS to skeletal system. That means to the voluntary fellow, right? To the voluntary fellow. Skeletal muscle is the muscle that is going to move, that is going to do any kind of movement, right? So it is going to be your skeletal muscles. Now autonomic, students, autonomic is going to send the impulses from CNS to visceral organs. That means your inside organs. Okay, visceral organs, or we can also write smooth muscles, because I have already told you this, that smooth muscle lines the visceral organs. Okay, smooth muscles. Smooth muscles. Clear? Okay, so these are your two systems again, autonomic nervous system and your somatic nervous system. Autonomic and somatic nervous system. Again, this is further divided. So here if we go back, okay, here you can see that the autonomic nervous system is further divided into sympathetic neural systems and parasympathetic neural systems. Visceral nervous system is a part of the peripheral nervous system that comprises the whole complex of nerves, fibers, ganglions, plexus, by which impulse travel from the central nervous system to the viscera. That means the organ, okay, the cavity where the smooth organs are there, or the visceral organs are there, from viscera to the central nervous system. Clear? So obviously visceral nervous system is going to do what, students? Obviously it's going to send signal to the visceral organs, right? The visceral organs. So same thing is going to happen here. Same thing is going to happen here. I hope so far it's clear. Now, neuron as a structural and functional unit of neural system. A neuron is a microscopic structure composed of three major parts, namely cell body, dendrites and axons. So as I told you, cell body, or you can also call it cyton, okay, you can also call it soma, right? These were the names I told you. Dendrites are the short branch structure. Branch structure is important to remember. Axon is a long process which is not at all branched. The cell body contains cytoplasm with the typical cell organelles and certain granular bodies called as Nissel's granules. Okay, short fibers which branch. Okay, Nissel's granules job is there, students, so that you have to remember. Okay. Now, the short fibers which branch repeatedly and project out of the cell body is also contains Nissel's granule and are called as dendrites. Okay, they are called as Nissel's granules and are called as dendrites. These fibers transmit impulses towards the cell body. So imagine one impulse is coming like this, it is going to transmit towards the cell body, then it is going to be carried all the way to the axon to the next dendrite, right? So students, what did they say? This is the branched portion called the dendrite. Okay, there is this Nissel's granule. Actually, this Nissel's granule, if you remember, they are going to synthesize a protein, okay, which is a neurotransmitter. Okay, a protein which is a neurotransmitter, and this neurotransmitter will be stored inside the axon knob, or we can tell the axon terminal. Okay, synaptic knob, right? Inside that it will be stored. Now look here, these fibers transmit impulse towards the cell body. The axon is a long fiber, okay, and the distal end of which is branched. Each branch terminates as a bulb-like structure called as the synaptic knob, which possesses synaptic vesicle containing the chemicals called as neurotransmitters. So the end of the axon, that is a bulb-like structure which is called as the synaptic knob, which will have the chemicals called as neurotransmitters, which are chemicals. Okay. 1 second, students. Okay, done. Okay, next one, this neurotransmitter. The axon transmits nerve impulse away from the cell body to a synapse, okay, or a neuromuscular junction. Based on the number of axon and dendrite, the neurons are divided into three types. Okay, multipolar, that means one axon with two or more dendrites, like this. Okay, they are found in the cerebral cortex. Okay, cerebral cortex. Bipolar means, students, one axon, one dendrite. Okay, what is it? Bipolar means one axon and one dendrite. That is there. Clear to all of you? Yes or no? So it could be something like this. Okay, I'll draw and show you later. With one axon, one dendrite, it will be bipolar, found in the retina of your eyes. Then you have unipolar, where there is a cell body with one axon only. Okay, cell body is there with one axon only. Okay, no dendrite, nothing is there, just cell body and axon. Got it? Chalo. Now, there are two types of axon, namely myelinated and non-myelinated. Myelinated means what? Ma'am, the ones who have myelin sheath. Non-myelinated means what? Ma'am, the one who doesn't have myelin sheath. Simple as that. Now, there is a set of cells called the Schwann cells. See, look here. Can you see the cells here? Schwann cells. These cells actually release the myelin sheath, which is a white colored substance. Okay, they release or they form this myelin sheath. See, the myelinated nerve fibers are enveloped with Schwann cells, which form a myelin sheath around the axon. The gaps between two adjacent, see, gaps between two adjacent myelin sheath is called as the nodes of Ranvier. Nodes of Ranvier. Here you can see. This is the portion where the axon will be exposed. Okay, this is the portion where the axon will be exposed. This question has come once in NEET, so you can remember this. Okay, you can always remember this. Okay, yes. Now the gaps between two adjacent myelin sheaths are called as nodes of Ranvier. We already understood, no? Myelin sheath, myelin sheath. This is going to be nodes of Ranvier. Okay, all of you quickly mark your attendance by hitting that like button and also subscribing to the channel. We are going to give you a lot of content on this channel as well, so definitely you can subscribe. And if you want long lectures and you want proper NEET level coaching, definitely follow us on Vedantu NEET English. That is our channel, which is called Vedantu NEET English. Okay, if any of you are looking for a pure English channel which is preparing you for competitive exams like NEET, then this is your channel, students, Vedantu NEET English. So definitely subscribe. And if you're a student who's looking for boards, okay ma'am, I want to learn NCERT level boards, then it is V Karnataka. Okay, please subscribe to this channel and make a note of the names as well. V Karnataka, KCET and boards. Okay, this is another channel. So this is our two channels that gives you all solid content every single day. Long marathons are taken there. Notes will be shared on the Telegram channel as well. So definitely follow us on the Telegram channel. It's Biology by Gopika if you want to follow me on Telegram channel. Now, the gaps between two adjacent myelin sheaths are called as nodes of Ranvier. Myelinated nerve fibers are found in the spinal and the cranial nerves. Remember I told you about these nerves? PNS has your spinal nerves as well as cranial nerves. That will have your myelinated nerve fibers. Myelinated means they will have myelin sheath. Okay. Unmyelinated nerve fibers is enclosed by a Schwann cell that does not form a myelin sheath around the axon, and it is commonly found in autonomous and somatic neural systems. Okay. Somatic neural systems. So let's go to the next page. Okay. I think all of you can see. Okay. So students, as mentioned, what and all did we study? We learned about your neurons. Okay, let's try drawing a neuron. It's actually hard to draw with the pen, students. Okay, but still I'm trying it for you guys. Yes, kind of okay. Okay, done. Okay, this is your neuron. Okay, I know some of you will be doubtful, ma'am, is this Pakka neuron or is it a dried tree that is there? Okay, so this is a neuron, and this what I'm drawing with your dark yellow is a dendrite. Okay, with the dark yellow is your dendrite, and in between there is your nucleus, and there is this dot structure that I'm drawing, which is your going to be a cell body. Okay, now students, in here there's a very important thing that you have to remember. There is granules, right? There is granules. Hello. So there are granules, and these granules are going to be very important for you to remember. Now let's say we will draw a myelinated neuron. Okay, this is your myelin sheath. Okay, this is your Schwann cells. Again, myelin sheath. This portion you will have Schwann cells. Myelin sheath. Okay, now student, this is your myelinated neuron. Okay, now let's start labeling and try to understand. So this is a short branch structure which is called the dendrite. Yes or no? It is a dendrite. Now this dendrite is there. After that, here this portion, what is there? This is your cell body. Okay, this is your cell body or cyton. Okay, cell body, cyton, perikaryon, anything you can tell. Then students, if you come here, this what you're seeing here is your myelin sheath. Okay, myelin sheath. And this myelin sheath is secreted by whom? It is by your Schwann cells. Schwann cells will secrete the myelin sheath. Now, between two myelin sheath, there is a space. Okay, that space is called nodes of Ranvier. Okay, nodes of Ranvier also we tell, Ranvier also we tell. Okay, because we have an actor named Ranvier, it will be easy for us to remember. So, the nodes of Ranvier is very important. This is the place where your axons are exposed. Okay, axons are exposed in this region. So do not forget this. Now when you come to the end, you will see synaptic bulbs. Okay, synaptic bulbs, or you can call it knobs. Okay, knobs. Inside the synaptic knob, it's going to be synaptic vesicle which will have the neurotransmitter. And this structure, unbranched log structure, is going to be called your axon. Now let's go to this blue color structure. This blue color structure is nothing but Nissel's granule. What is it? Nissel's granules. And this Nissel's granule, students, is important because it will be synthesizing. It can synthesize a protein, and that protein will be a neurotransmitter. Okay, that protein will be a neurotransmitter. Okay, and it will be stored in the synaptic vesicle. Stored in the synaptic vesicles. Got it, all of you? Yes or no? Clear? Okay. No one is marking the attendance, guys. What happened? Clear, all of you? Yes or no? Tell me quickly. Clear? Okay, fine. Okay, then let's go back. Okay, let's go back and let's try to learn the second part. Now students, this much they have told. Now you have to understand few more things, like, okay, like this is your synaptic knob. Okay, at the end of the axon. Okay, synaptic knob at the end of the axon. Now if you see inside the synaptic knob, there are okay, let's say inside the synaptic knob there are synaptic vesicles. Okay, inside the synaptic vesicles there is going to be your neurotransmitters. Okay, there is going to be your, this color is not visible, neurotransmitters. Okay, and one of the neurotransmitter will be synthesized by your Nissel's granule, which is present in the cell body, which is present in the dendrite, but not present in the axon. This question also has been asked previously in NEET. Okay, so this is the synaptic knob. Okay, or vesicle, knob. Now this is the vesicle. Inside that there is a storage, no, like that we did. So it's a synaptic vesicles. Now inside this vesicles there is neurotransmitter. So during chemical synapse, this neurotransmitter will be released. During chemical synapse or chemical impulses. Clear? Yes or no? Chalo. Okay. Now we'll come here. Okay, I have to teach you this also, no? Types of neuron. Okay, I'll teach you types of neuron. Just give me 1 minute. Okay. Now, types of neurons, students. Types of neuron. Depending on, how is this types of neuron done? Depending on the number of axons, okay, number of axons and number of dendrites. Okay, number of axons and number of dendrites, they have done the classification. And classification, first one is going to be unipolar. Okay, first one is going to be unipolar. Unipolar means what do they indicate by unipolar? There was one axon and one cell body. Bus, that's all. Okay, that's all. One axon, one cell body. Ho gaya, kaam ho gaya. No? So this is the cell body. Okay, cell body. And this is the axon. Got it? Ho gaya. Done. Job is done. This is unipolar. This is unipolar. Now, bipolar. Okay, bipolar. They meant by bipolar what? That means one axon is there, one dendrite is there, one cell body is there. So, one cell body. Okay, one dendrite. Just one dendrite. Okay, and one axon. One axon. This is called as bipolar. Okay, one dendrite, one axon, one cell body. Everything is one. Okay, next one is going to be your multipolar, which you are seeing normally. Okay, multipolar is the thing the ones you have seen, students. This one axon and many dendrites. Okay, they have given 2 or more dendrites, but let's just assume more dendrites. Okay, more dendrites are there. Check, and here. This is your multipolar. Then there is one called pseudo unipolar. Okay, there is one more called pseudo unipolar. Pseudo unipolar. Pseudo unipolar. Pseudo unipolar means, students, halfway this fellow told that, oh, it looked like this fellow is going to become unipolar. Like he started like this. He had one cell body. Okay, he was emerging like this. Everyone thought, oh, okay, pakka, this fellow is going to become unipolar. Okay, pakka going to become unipolar. He started like this. He started emerging like this. After he emerged for some distance, he became a partition. He started to partition. That is when people understood, oh, this fellow was lying. He is not going to be unipolar. He is pseudo unipolar. Because look at unipolar, one cell body, one axon. Okay, that is how it was going to form. Later, he suddenly branched to forming some dendrite or something. So he became pseudo unipolar. Understood? Now, where are these people present is a question that will come in NEET as well as in your boards. Where are these people present? Now, unipolar, what is the answer, students? Where are they present? They are usually present in lower organisms. Okay, they are usually present in lower organisms and invertebrates. Now, bipolar is seen in the retina of our eyes. Okay, it is seen in the retina of eyes. This multipolar is seen in the cerebral cortex. Okay, cerebral cortex. And pseudo unipolar is usually found in the dorsal root of the spinal cord. Dorsal root ganglion of spinal cord. Understood? You should remember the location. If you do not remember the location, then it's going to be a bad thing, because it's going to be actually difficult for you to learn it. Okay, difficult for you to learn it. Chalo, let's move forward. This whole paragraph I've made you write, and also I've made you explain. Now, generation. Okay, generation of conduction of impulses. Okay, let's go. Here, Neurons are excitable cells because their membranes are in polarized state. Now, what is polarized state? That means in a stable situation, we tell, no, like stable or both of them are equal. Okay, so here's polarized state. Do you know why the membrane of the neuron is polarized? Different types of ion channels are present on the neural membrane. These ions are selectively permeable to different ions. When a neuron is not conducting any impulses, that is during the resting situation, that means no impulses there, everyone are relaxing, it's a total relaxed situation, that is a resting situation. The axon membrane is comparatively, student, this is a very important line from NCERT point of view, from your board point of view, from a NEET point of view. This is a very important line. Okay, this is a most important line, that the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. Okay, they'll be more permeable to potassium ions and they'll be impermeable to sodium ions. Similarly, the membrane is impermeable to negatively charged proteins present in the axoplasm. Consequently, the axoplasm inside the axon contains high concentration of potassium and negatively charged proteins and low concentration of sodium. In contrast, the fluid outside the axon contains a low concentration of potassium and high concentration of sodium. Now, students, here what are they talking about? They're talking about the concentration. No. So, let's say if I draw, this is the axon. Okay, this is the membrane. Okay, now on the top portion, okay, on the upper side or on the, we can tell, the outer fluid, we have positive side. Okay, positive. Why is this positive sign there? It is because of the presence of sodium. Okay, it's because of the presence of sodium. So let's say in the extracellular fluid, okay, extracellular fluid, we have lot of sodium ions. Okay, lot of sodium ions, and that's why we have the positive sign. Now, at the same way, on the other end, okay, on the other end, let's say we have the negative. Now, students, this negative is not because of the presence of potassium, but it is because of the presence of negatively charged protein. Okay, negatively charged protein. They are responsible for this negative charge, but with them, potassium also is present. Okay, potassium also is present. Now, what are they telling? They are telling that this membrane is more permeable to potassium ions than the sodium ions. Okay, now when an action potential is created, okay, that means something has happened or an action potential has been created. That time, what is going to happen, students? Very simple. Charges are going to interchange. That means sodium will flow from a region of higher concentration to lower concentration. That means sodium will start moving inside. Okay, let's so this is your extracellular fluid now. Okay, say this is extra, this is intra. Okay, this is extracellular fluid. Okay, and this is going to be intracellular fluid. So interchanging is going to happen. That is what they're talking about here. So listen to me, listen to this carefully. Do you know why the membranes? Okay, why the membranes? This axonal membrane is comparatively more permeable to potassium ions. This is clear, no? Now nearly impermeable to sodium ions. Similarly, the membrane is impermeable to negatively charged protein. So inside, okay, inside the membrane, as I told you, negative sign is there, no? That negative sign is because of the negatively charged proteins. Students, I cannot write very well because of the pen. Okay, remember that. I'm writing it on the this one. Negatively charged protein. So it is going to be impermeable to. What, Shubham? Okay. Now, negatively charged proteins are low. In contrast, the fluid outside, okay, remember like this, students, two layers are there. Okay, this is the axon layer, this is one layer, this is down one layer. What are they telling? They are telling that, okay, let's fold this. Okay, let's fold this. Okay, this is the membrane. Okay, this is the membrane. Let's say here positive is there, here negative is there. Now, what are they telling? They are telling that the fluid outside the axon contains low concentration of potassium. Yes or no? Here potassium is going to be more. This is inside solution. Here potassium is going to be very, very less. Right? Potassium is going to be less. Now, a high concentration of sodium thus forms a concentration gradient. These ionic gradients across the resting membrane are maintained by active transport of ions by sodium-potassium pump. Okay? Sodium-potassium pump. So, here we can understand to regulate these people, okay, to regulate this proper movement of these ion, there is a sodium potassium pump that is present. Okay, NaK pump is present. Clear to all of you, right? No doubts, right? And students, these people have leaky channels. So if I have to draw, okay, if I have to draw like this, they have leaky channels. Okay, these, let's say these are the openings, these are the openings, like this. Okay, leaky channels will be present. Same thing goes here also. Through this leaky channel, sodium and potassium exchange is going to happen. Okay, yes, hi Mahi. Okay, exchange is going to happen. Perfect. Okay, perfect. Now one second, yeah. Okay, this is so far good, no? So they will have, students, usually axolemma, okay, axolemma will have up to 2 leaky channels. Okay, 2 leaky channels, and they will have 1 pump. Okay, 1 pump, which is sodium potassium pump. Okay, sodium potassium pump. Clear? So far good? Hi. Those who are telling hi. Okay, now remember that whenever energy, this is the energy process. Okay, this is going to be an energy process. Okay, I want all of you to like the video. All of you like the video, click on the like button. Okay, now students, what is going to happen when 1 ATP, okay, when we are going to use 1 ATP, when we use 1 ATP, 1 ATP is equal to 3 sodium, okay, 3 sodium ions will be kicked out, okay, kicked out, and 2 potassium ions will be kicked in. That means 2 potassium ions will be taken in. I'll tell you how to remember this, okay. There is actually one example for this. I'll tell you later, but right now try to understand. Okay, now imagine, students, exchange has happened. Okay, so first this situation will happen. Okay, first all the positive sign. This is resting situation. Okay, we'll draw one by one. This is resting. This is resting. Okay, from resting situation, one second. Okay, done. Okay, now students, look here. Imagine at the first situation, sodium starts coming in. Okay, so sodium starts coming in. Let's just draw like this. Okay, let's just draw like this. Sodium has started coming in. Okay, so plus signs, plus sign, plus sign, plus sign, plus sign, plus sign, plus sign. That means from outside the sodium has started moving inside. Here it is still negative sign. Okay, here it is still going to be negative sign. No problem in that. Potassium hasn't started going that side. But after some time, when the threshold will be reached, potassium also will get angry. No, potassium will start going inside. So the mix of charges will happen. Now, if I want to make them again back to the resting potential, I am going to use the pump, and that pump, okay, is going to use ATP and make sure that 3 sodium ions go outside and 2 potassium ions come inside. Why? To maintain the balance. Okay, this is a little difficult concept, but if you understand once, then you're sorted. Okay, if you understand once, then it is easy for you. Yes or no? Clear? Okay. Now, this you remember always, 1 ATP can remove 3 sodium ions outside and 2 potassium ions it can take inside. Now, how does an impulse happen, students? Let's say here one impulse happened, okay, which is around 55 minus 55 to 65 MV. Okay, now I'll go back to NCERT and make you read. See, thus the concept, these ionic gradients across the resting membrane are maintained by active transport of ions by the sodium potassium pump, which transports 3 potassium outwards, okay, 3 potassium outwards and 2 sodium ions into the cell. Okay, so in the extracellular we need sodium, intracellular we need potassium. As a result, the outer surface of the axonal membrane possesses a positive charge, yes or no, while the inner surface is going to have the negative charge. Okay, negative charge. Okay, now look at this diagram. We'll try to understand from the diagram. So students, this is the resting potential. Can you see positive, positive, positive outside, negative, negative, negative inside? Suddenly, one impulse has come here. Okay, from the resting potential, impulse has come. So, the sodium will start moving inside. Okay, from both the sides, sodium will start moving inside. So, you can see positive signs have been achieved here and negative signs have gone outside. Okay, now this process is going to continue, right? So, here you can see the signs are going to move this way. That is repolarization, depolarization is going to happen. That I'll tell you in sometime. I'm going to tell you that in some time. Okay. Now listen carefully. The outside of the membrane, will become negatively charged and therefore is polarized. The electrical potential difference across the resting plasma membrane is called as the resting potential. You might be curious to know the mechanism of generation of nerve impulse and its conduction along the axon. So from here you have to listen carefully. When a stimulus is applied to a site, okay, when a stimulus is applied to a site, let's say in one site stimulus has come, okay, that is Praveen is touched a hot object, okay, Praveen touched a hot object and Praveen's hand might burn. So that time an impulse has come. So normally how it is in resting potential, no? So this is how the sign is going to be positive, 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 positive. Okay, inside it's going to be negative, 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 negative. Now let's see what they're trying to say. When a stimulus is applied on a site, okay, this is the stimulus or the impulse you can write. Okay, stimulus or impulse. Okay. On the polarized membrane, the membrane at site A becomes freely permeable to sodium. So here they will become freely permeable to sodium. So here let's say this leads to freely permeable to sodium. This leads to rapid influx of sodium ions followed by reversal of polarity at that site. So this leads to what, students? Next thing that you're going to see is outside it is going to be negative. Not fully, okay, one by one, slowly it will change. Slowly it will change. And here it is going to be positive. Can you understood? Why? Because when an impulses come, okay, when an impulse come, that portion is going to become permeable to sodium. Okay, permeable to sodium. And sodium will start going inside. Okay, it's very simple. Just region of a higher region to lower region. Now, the outer surface of the membrane becomes negatively charged. See, outer surface became negatively charged and the inner side became positively charged. So far we are following, okay. The polarity of the membrane at site A. Now, this is, let's say this is site A. Okay, this is site A. The polarity of the membrane at site A is thus reversed and hence depolarized. Okay. So students, what is going to happen? This is going to continue and this will shift this side and this will shift this side. Yes or no? The positive will move this way. Okay, and the negative will move this way. So A to B and B to C. I have to just check that. Okay, A to B and B to C. Yeah. Here ulta. So here you can see, students, this will move this side. Okay, this will move this side and this will move this side. Okay, so A, let's say this is the B region. Okay, this is the B region. So this is going to continue. Okay, this is going to continue. Now look here. The electrical potential difference across the plasma membrane at site A is called as the action potential, which is in fact termed as the nerve impulse. Okay, this is the action potential created. So same thing only you are seeing here in the diagram. Look at the diagram here. You will notice. Okay, what are you seeing here? First, you will see that outside negative sign has come, inside positive sign has come. Slowly, you will start noticing that this positive sign is moving forward. Okay, so in the outside the membrane, it is going to move forward, and inside the membrane, it is going to move backwards. Okay, going to move backwards. Students, only first time when you learn, this is going to be difficult. Second time and all, it will be very easy. Okay, and if you want a proper lecture of this, you can check Neural Control and Coordination by Gopika ma'am. At the site immediately ahead, the axon membrane has positive charge. See, at the site immediately front, you have positive charge, and on the outer surface, you will have negative charge. Okay. As the result, a current flows on the inner surface. Very important, students. Inner surface from A to B. Okay, here it is, say, it's too congested, no? In the inner surface, this is the A region. Okay, this is the A region, so it will move like this, A to B. Okay, A to B, this is important. And on the outer surface, it is going to go from B to A. Okay, outer surface it is going to go from B to A. This is what they have told. This is what they have told. Done. Done. A to B, B to A. So inside inner surface, A to B, outer surface, B to A. Okay, B to A. Clear? Okay, perfect. Okay. Now, on the outer surface, the current flows from B to A side, complete the circuit of the current flow. Hence, the polarity at the site is reversed and the action potential is generated at site B. Thus, the impulse generated at site A arrives at the site B. The sequence is repeated along the length of the axon, and consequently, the impulse is conducted. The rise in the stimulus-induced permeability to sodium is extremely short-lived. It is quickly followed by the rise in permeability of potassium. Within a fraction of seconds, potassium diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation. I think. Okay. The transmission of impulse, we will do it after a break. See you after break, students. Yes, let's continue, students. What happened to this? Okay, now we have done transmission, right? Saltatory conduction. I think I'll come to that. Okay, so here we understood how polarization is going to happen. Now transmission of impulses. So students, remember that when an electron, this is a neuron, this is a neuron, if the gap, okay, if the gap between two neurons, okay, if the gap between two neurons is going to be less than 2 nanometer, okay, a chemical synapse is going to happen. Okay, gap is more than 2 nanometer, a chemical synapse is going to happen. Chemical synapse means what? We are going to use a neurotransmitter for the job. Okay, we are going to use a neurotransmitter for the job. Clear or not this is? Okay, chalo. Okay, clear? Okay. Now look here, what is a synapse? Let's see that. Something happened, students, I'm so sorry. Okay, a nerve impulse is transmitted from one neuron to another through a junction called synapse. A synapse is formed by membranes of presynaptic neurons and postsynaptic neurons, which may or may not be separated by a gap called synaptic cleft. Okay, so two neurons are there. Hello, hello. Okay, two neurons are there. What is a synapse? Now let's say this is a neuron, this is the dendrite, this is the axon. Okay, this is the axon. Same way here, this is the dendrite, this is an axon. So it is a head to tail connection. Okay, head to tail connection always. So this dendrite is going to connect to this head. Now look here, a nerve impulse is transmitted from one neuron to another through a junction called synapse. A synapse is formed by the membrane of a presynaptic neuron. Okay, membrane of a presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap. There are two types of synapses, namely electrical synapses and chemical synapses. At electrical synapse, the membranes excuse me, so sorry students, again Bangalore weather is changing every 5 minutes, so it's very difficult to survive here okay, with the climate. Okay, chalo. There are two types of synapse, namely electrical synapse and chemical synapse. At electrical synapse, the membranes, okay, the membranes of pre and post synaptic neurons are in very close proximity. So here what are they telling? In an electrical synapse, the membranes of your pre, this is your pre, okay, this is your post, they both are going to be in close proximity. Okay, they are going to be in close proximity. Why they are in close proximity? Because the signal has to be sent. It's an electrical impulse, right? Neurotransmitters means there is a synaptic lobe. Here there is neurotransmitter. That is a different story. Okay, this is going to happen because electrical impulse, electricity types, you know, same way impulse is going, it has to go from pre to post. They both have to be in close connection with each other. Now electrical current, okay, electrical current can flow directly from one neuron into another through these synapses. Transmission of an impulse across an electrical synapse is very similar to the impulse conduction along a single axon. Okay, along a single axon. Now, impulse transmission across an electrical synapse is always faster. Okay, impulse transmission across an electrical synapse is always faster than that across a chemical synapse. So obviously students, now say a person is walking and trying to deliver, and then there is a person who is coming in a Zepto vehicle and delivering. Who is going to reach faster? Obviously the person who went in a vehicle, right? Same thing here also. Electrical synapse is always going to be faster than a chemical synapse. Okay, electrical synapse are rare in our systems. Okay, so electrical synapse are comparatively rare in our system. At a chemical synapse, the membranes of the postsynaptic neurons are transmitted by a fluid space called synaptic cleft. Okay, hi Roshni. Okay, filled space called a synaptic cleft. Now, do you know how the presynaptic neuron transmits an impulse across a synaptic cleft and a postsynaptic? That we have to see. Okay, that we have to see. See, chemicals called neurotransmitters are involved in the transmission of impulses at the synapse. The axon terminal contains vesicles filled with these neurotransmitters. When an impulse or an action potential arrives at the axon terminal, it stimulates the movement of synaptic vesicles towards the membrane where they fuse with the plasma. So students, look here. This is a beautiful diagram. Look here. This is an axon. Okay. As I told you, this is your axon. Okay. It might look like chicken feet, but it is not. Okay, boom, this is your axon. Got it? This is your axon. This knob, this portion is enlarged here. So look here, axon terminal. Inside that, I told you synaptic vesicles will be there. Inside the synaptic vesicle, the yellow color thing that you're seeing is a neurotransmitter. Okay, now this is your presynaptic, this is your postsynaptic. So post, between this, what is there? That is the synaptic cleft. So try to understand, this is your presynaptic. Okay, presynaptic. This is going to be a synaptic cleft, and this is going to be postsynaptic. That is actually a portion of the dendrite. Okay, this also we actually draw like this. So the portion of the dendrite. Got it? Like this. So this becomes pre, this becomes post, this becomes synaptic cleft. Got it? Now this is the presynaptic. Okay ma'am, this is the synaptic vesicle. Inside the neurotransmitters are there, they will be released, and here you can see on the postsynaptic cleft there will be receptors. Okay, receptors waiting to collect this neurotransmitter. Okay, receptor. So these neurotransmitters will be here. Okay, they will open like this. Okay, they'll open like this, they'll get released, they'll go and be collected by the receptors. Yes, very good. Now these receptors will collect it, and definitely the rest of the job they are going to take care. Now see here, the released neurotransmitters bind to their specific receptors present on the postsynaptic membrane. This binding opens ion channels, allowing the entry of ions, which can generate a new potential in the postsynaptic neuron. The new potential developed may be neither excitatory or inhibitory. Excitatory means that is going to continue the reaction. Okay, inhibitory means that is going to stop the reaction. Got it, all of you? Clear or not so far? Yes. Okay, let's say. So it's going to be something like this. This is your presynaptic cleft. Okay, oh, like this. Okay, this is your post. Okay. Ma'am, recently I came to know that a person can live without pituitary gland provided they take hormone replacement, but they will be normal. See, Roshni, like we can do hormone replacement, injecting the hormone, but the problem is, normally what our body does, if you try to do artificially, it will not be proper. Okay, it will not be proper. So that definitely there is going to be a drawback is definitely going to be there. Okay, got it? Yes, is this clear? I think research is still going on ADHD meditation and how it is going to work. Okay, so there is going to be a drawback to it, but we will not know exactly how the drawback is going to come. It could be because of a release. See, because pituitary is a master gland, it controls all other glands. So that doing with the help of an external thing is going to be a problem. Okay, so I will definitely read a lot more about it. Okay, so and then I will give you a clarity on the same. Now students, this is your synaptic vesicles which have opened. Okay, This were your synaptic vesicles previously. Inside this they were there. Okay, here again they will open. Okay, on the impulse coming they'll open, and inside this the neurotransmitters are there. Okay, I'll try to edit. Oh no, it vanished. Okay, let's draw it again. Okay, this is the presynaptic. So students, inside this neurotransmitter will be there. It could be acetylcholine, it could be dopamine. Okay, it could be any of that. Now here see the neurotransmitter got released, and there will be receptors here. Okay, there will be receptors here waiting to collect these neurotransmitters and act accordingly. Okay, receptors. So this is how the system works. If you want, I'll label this presynaptic, synaptic vesicle. Okay, synaptic vesicle. Okay, and Ratis, where were you for the evolution class? I did not see you. Where were you for the evolution class? I spoke about you people in the class also. Roshni also I spoke about. Ratis also I spoke about. Okay, neurotransmitter. This is going to be your post synaptic cleft. Okay, we can call it a cleft or post synaptic portion. Clear? Yes. Now this neurotransmitters, two things is there. This neurotransmitters can be inhibitory. Transmitters can be inhibitory or excitate. Got it? Yes. Clear? The way I am writing or drawing is like I'm staring into space, but no students, I'm not doing that. Okay, if you want, I'll give you some examples of excitatory ones. It means who is going to continue the reaction and who is going to stop the reaction. Okay, now acetylcholine, ACH, clear ma'am. Okay, epinephrine. These two examples you can remember. Inhibitory students, you can remember dopamine. Okay, you can remember serotonin. Yes, GABA also. Okay, done. So far good. Now the central neural system or the central nervous system. Okay, the brain is the central information processing organ of our body and acts as a command and control system. It controls the voluntary, movements, balance the body function and functioning of the vital involuntary organs. Thermoregulation, hunger, thirst, circadian rhythm, everything is controlled by your central neural system. Okay, central neural systems. Now, it also is a site for processing of vision, hearing, speech, memory, intelligence, emotions, and thoughts. The human brain is well protected by a skull. Inside the skull, the brain is covered by cranial meninges. With what? Cranial meninges. I have shared this on my Telegram. If you want, you can check. Okay, it's Biology by Gopika. On the Telegram, I have shared you how the cranial meninges are going to look like. Okay, I have shared about the dura mater. Yes, they consist of an outer layer. So students, if you have to see this, okay, so you have dura mater at the first layer, then you have middle layer arachnoid, inner layer you have pie mater. So you remember DAP, that is from outside to inside. Outside to, this question has come, so remember this, outside to inside it's going to be DAP. Okay, inside to outside, it's going to be pad. That is pie matter, arachnoid, dura matter. The brain can be divided into three major parts: forebrain, midbrain, and hindbrain. This who doesn't know? Who doesn't know? Okay, forebrain, midbrain, and hindbrain. See, look at this. This is your brain. Okay, this is how your brain looks. See, cerebral hemispheres, you have studied about it. Corpus callosum also, I think all of you would have studied. It's the joining portion. Then cerebral aqueduct, which you are going to study, a part of your midbrain. That is going to be new for all of you. Okay, new for all of you. Then you have pons, cerebellum, medulla. This is going to be a part of your hindbrain. Midbrain is here. Forebrain is your cerebrum, thalamus, and hypothalamus. Okay, this is going to be the portion of your brain. Okay. Rhinovirus loves Gopikama. Okay, how much you more than you people. Whenever you show less love, rhinovirus will come and attack. For evolution class, you people showed less love. No, that's why I got caught by rhinovirus. Yesterday KCET channel pe also you people did not come. No, that's why I got rhinovirus. So whenever my students are showing less love, they're not attending class enough, rhinovirus will come. Students, I don't know what's happening. This is working by itself. Okay, this is better. Okay, the forebrain consists. Yeah, I have been hearing this a lot now. Okay, I look like Dr. Momita. Yes, cerebrum, thalamus, and hypothalamus. The forebrain consists of cerebrum, thalamus, and hypothalamus. Cerebrum forms a major part of the human brain. A deep cleft divides the cerebrum longitudinally into two halves, which are termed as the left and the right cerebral hemispheres. The hemispheres are connected by yes, Aman, I have heard this a lot. Okay, now I have heard this. Yeah, it's because of the cheeks. I have heard that she and we both resemble. But yeah, let's pray for her soul. Okay, the forelimb consists of cerebrum and thalamus and hypothalamus. Cerebrum forms the major part of the human brain. A deep cleft divides the cerebrum longitudinally into two halves: right cerebral hemisphere, left cerebral hemisphere, and these hemispheres are connected by corpus callosum. Okay, this is what I told you. There is a connection. Okay, corpus callosum. The layer of the cell which covers the cerebral hemisphere is called the cerebral cortex and is thrown into prominent folds. Okay, chuk 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 chuk, like this. Brain you have seen, no? So let's put brain emoji in the chat. Like this, folds are there, no? Many folds are there. Many folds are there, no? That is how it is. Okay, the cerebral cortex is referred to as the grey matter due to its greyish appearance. The neuron cell bodies are concentrated here, giving the, so grey matter is because of the cell body. Okay, and the white matter is because of the axon. Why? Because the axon has a white myelin sheath. Yes or no? Axon has a white myelin sheath. So obviously they are going to have white color surface. The neuron cell bodies are connected here, giving the color. The cerebral cortex contains motor areas, sensory areas, and large regions that are neither clearly sensory nor motor in region. These regions are called as association areas. So student, there is one motor neuron. This I think you have studied in smaller classes. Motor neuron, okay. Then you have sensory neuron. Okay. Then you have association neuron. Okay. Yes. Now let's go again. So responsible for complex functions like intersensory association, okay, memory and communication. Fibers of the tracts are covered with myelin sheath, which constitute the inner part of the cerebral hemisphere. They given an opaque white appearance. Okay. Yes, very good, Aman. You're preparing for NEET from 10th standard. Very good. Okay, great. Now you should definitely be Air 1. Okay, definitely be Air 1 because you're preparing for almost 3 years. Okay. They give an opaque white appearance to the layers, and hence it is called the white matter. White matter is because of the presence of axon. Okay, because of the presence of axon. So students, so far you have learned 2-3 things. Okay, first thing you have to remember is your brain. Oh, okay, I'll write it here. Like this. Brain has 3 portions. Okay, forebrain, midbrain, and hindbrain. Okay, hindbrain. Yes or no? Hindbrain. Correct? Okay. Now, forebrain. Okay, we didn't come to that. Now, let's say brain has covering. Okay, that is called as cranium. That is the skull. Okay, they have meninges. Those meninges are called as your dura mater, arachnoid, pie matter. Okay, pie matter. Got it? Now, now students, if you have to remember from out to inside, okay, out to in, it is called DAP. Okay, in to out, it is called pad. Okay, that is how you have to remember. This question was being asked in NEET. Clear or not? Visible to all of you? Visible or not? Okay, done. Now we go back and you continue here. See, what are they telling? They're telling that another very important part in the brain is called your hypothalamus, who is the principal. If pituitary gland is the master gland, then hypothalamus is the principal. Okay, it lies at the base of the thalamus. The hypothalamus contains a number of centers which controls the body temperature, okay, urge for eating, drinking. It also contains several groups of neurosecretory cells, which I will be teaching you in chemical coordination. Okay, which I'll be teaching you in chemical coordination. Yes. Now, which secretes hormones called hypothalamic hormones. Okay, what are they going to release? Hypothalamic hormones. The inner part of the cerebral hemispheres and the group of associated deep structures like your amygdala and hippocampus form a complex structure called the lymphic lobe, which is responsible for sexual behaviors, expression of emotions, excitement, pleasure, rage, fear, motivation. Students, this is not working for you people. Understood? Your limbic lobe is not working for all of you. Okay, you found the problem. Now you don't have to go to a doctor. You can understand why you don't have motivation to study and crack that exam that you have been dreaming from a very young age. Is because your limbic lobe is not working. Okay, motivation is not there. You people tell no every time. See, this is the reason. Motivation is not there because limbic lobe is not working. Kho gaye hum kahan. So that is the situation, right? Yeah. Now midbrain. Ready for midbrain? Okay, the midbrain is located between thalamus and hypothalamus of the forebrain and pons of the hindbrain. Okay, so I have become a mini doctor version. So see, I am telling you which is not working for you people. Okay, yes or no? Okay. Fine. Now midbrain, students. The midbrain is located between the hypothalamus and thalamus of the forebrain pons of the hindbrain. A canal called the cerebral aqueduct passes through the midbrain. Okay? Yes or no? You have to nod. You have to recharge it. You have to recharge it and make it work. Chumma, it will not work. Okay? So long you have kept it silently. How will it work suddenly? You should also think about it, no? That, oh, Pavam, he's not working. Oiling, to oil it is not some gear or cycle or something, no, that it will start working immediately. So that's the problem. Okay, so students, the canal called the cerebral aqueduct passes through the midbrain. The dorsal portion of the midbrain consists of main 4 round Okay, so this, this, and this. These are your corpora quadrigemina. Okay, corpora quadrigemina. This corpora quadrigemina, students, is going to control with your vision. Okay, let me draw it here. Yeah. Okay, it is going to be helping with your visual and auditory reflexes. Is this clear? Okay, now you can draw a structure like this, and you can draw a structure like this. Okay, this is your corpora quadrigemina. They are going to help with your visual reflexes as well as your auditory reflexes. Okay, so this is called as your superior colliculi, and this is going to be inferior. Colliculi, inferior colliculi. Clear or not? Inferior is going to take care of the auditory reflexes and superior is going to take care of the visual reflexes. Now, hindbrain comprises of pons, cerebellum, medulla, and it is also called as medulla oblongata. Pons consist of fiber tracts that interconnects different regions of the brain. Cerebellum has a very convoluted surface in order to provide the additional space for many more neurons. The medulla of the brain is connected to the spinal cord. The medulla contains centers which control respiration, cardiovascular reflexes, as well as gastric secretions. Students, this is very important. 3 major regions make up the brain stem, that is called as your midbrain, pons, and your medulla oblongata. This is very, very important. Okay, this is very, very important. Brain stem is going to be constituted by your midbrain, midbrain, pons, and medulla oblongata. So now what did we study? Let's do a quick recap. Forebrain, we have studied that cerebral hemispheres are there, hypothalamus is there, and thalamus is there. Okay, so below thalamus, hypothalamus is going to be there, and they are going to have hypothalamic hormone secretions. Okay, now cerebral hemispheres are going to take care of all your intelligence. Okay, IQ level, everything is going to be taken care by your cerebral hemispheres. So far good. Now midbrain, when you come to the midbrain, you have cerebral aqueduct, which is a canal passing through the midbrain, and then you have corpora quadrigemina, which are 4 round swellings. Okay, these 4 round swellings can also be called as lobes. The top bigger ones are called as superior lobes, the down ones are called as inferior ones. Now the superior ones you call it as superior colliculi, and inferior ones you call it as inferior colliculi. So far good. Okay, chalo. Now the midbrain, when you come to, you have pons, cerebellum, and medulla. Pons is a portion, we will just keep it there because right now we're not talking much about pons only. Cerebellum is the portion that is going to take care of your balance. So when a person drinks alcohol, it is noticed that that person works zigzag pattern. Why does the person works in a zigzag pattern? Because the cerebellum is affected when a person is drinking. So cerebellum is going to maintain your balance. Now medulla is at the other place is going to take care of your breathing, is going to take care about your cardiovascular reflexes, gastric secretions, the tendency to vomit. All this is taken care by your medulla oblongata. Now in breathing and exchange, we have studied that these people have your pneumotactic centers, chemosensitive areas, and those portions are going to actually help in respiration. Okay. Now then, what did we study prior? Students, we studied in the prior that there is, meninges. Yes, if you come here, okay, if you come here, we have learned that your entire central nervous system. So previously we learned that your entire neural system or your nervous system is divided into central nervous system and peripheral nervous system. Central nervous system is further divided into brain and the spinal cord. Okay, peripheral nervous system is divided into spinal nerves, cranial nerves, or afferent fibers and efferent fibers. Afferent means the person who is bringing the blood. Okay, like that, like afferent we tell in glomerulus, bringing. So same way, afferent means the person who is bringing the information from the tissues to the CNS. The person who is going to take the regulatory CNS from the CNS, if it's going to take the information to my tissue, then that person is going to be called my efferent fibers. Okay, efferent fibers. Yeah, the creative walking is because their cerebellum is also walking inside. Okay, yes. Now students, so much we understood that the CNS, PNS is the PNS is again divided into somatic and autonomic. Somatic nervous system will send all the signals from the CNS to the skeletal muscles, and your PNS is going to, your autonomic nervous system is going to send all the signals to your visceral as well as your smooth muscles. Okay. Now, visceral system is there, which is a part of a peripheral nervous system. That fellow is going to, that system is going to send all the signals to the viscera. That means the cavity where your visceral organs are going to be there. This is how much we have learned. Now, when we talk about specifically central nervous system, we have to talk about brain and the spinal cord. Brain is covered in a cranial meninges, and that cranial meninges are three of them: dura mater, arachnoid, and pi mater. DAP is outside to inside, pad is inside to outside. Okay, pad is inside to outside. Now, brain is divided into forebrain, midbrain, and hindbrain. Forebrain we discussed, midbrain we discussed, and hindbrain we discussed. Clear? So far, good? This is not there for you guys. Okay, reflex arc is not there, eye is not there, ear is not there. All this is not there for you. Yes, none of this is there. So we have almost completed. Obviously, I have not taught you in detail. I have taught you just NCERT level. But if you want it for their NEET preparation, definitely go to Vedantu NEET English, search Neural Control and Coordination by Gopika ma'am, and you will get a detailed lecture for the NEET students. Okay, this is just for the board students, just to remember the main topics, what are the main terms and other things. But for the NEET students, I have taught transmission in a different way. I have given a lot of examples. I have taught you how to, understand that when depolarization happens, repolarization happens. So that and all I have not discussed here. Okay, this is just from your NCERT level. So there I have taught you everything based on your NCERT, that is your both in a detailed manner. Okay, in a depth manner I have taught you, where I have taught you about depolarization, repolarization, polarization, resting potential, action potential. Here that much is not discussed in the paragraph, but when we teachers teach, we usually teach extra. Okay, yes, clear or not, all of you? Perfect. Okay, yes. Yes, so that's it for today's class, students. See you all in the next class. Thank you so much for joining. See you all. Tata, bye-bye. If you want to join my Telegram, it's Biology by Gopika. And if you're looking for a NEET channel where you can get authentic content for NEET in detail, that is Vedantu NEET English. And if you're a board student who's looking for a channel, that is B Karnataka, KCAT and boards, where we are preparing you for boards. So thank you so much for joining. See you all in next class.